Hello and welcome. Today we're going to become exam detectives. We've got our hands on a full 17-mark question from an AQA GCSE Biology paper. But more importantly, we've got the official mark scheme. Our mission? To crack the code and see exactly what you need to write to get every single mark. Ready? Let's dive in.
So here's the plan. We'll start with how we're going to crack this exam code. Then we'll break down the question piece by piece, looking at the core photosynthesis experiment, the super important role of chlorophyll, and how to deal with data on rates and limiting factors. We'll even take a quick look at some physics with the law of light, and finally, we'll see how it all adds up to your final marks.
And that subtitle says it all, really. Getting top marks isn't just about knowing stuff, it's about showing what you know in the exact way the examiner wants to see it. This whole explainer is about mastering that technique by getting a little peek inside the mind of the mark scheme.
Right, let's get stuck into the first part of the question. This is a classic experiment, you've probably seen it before. It's designed to test if you understand the essential ingredients for photosynthesis. Let's see the setup.
Okay, so the exam gives you this table. We've got 3 leaves from the same plant. Leaf 1 is totally covered in black paper, so no light. Leaf 2 is sealed in a clear plastic bag, so it gets light but no fresh air. And leaf 3 is just left alone, our control. After 2 days, only leaf 3 has any starch. So, what's the question they're going to ask?
And there it is, a hefty 5-mark question. And the command word is explain. That's a huge clue. It means you have to give the reasons why things happen. So, how do we make sure we get all 5 of those marks? And here they are, the 5 golden tickets, straight from the mark scheme. Notice how specific they are. Mark 1, for leaf 1, you have to say there's no light, so no photosynthesis. Bam! Mark 2, for leaf 2, no carbon dioxide, so no photosynthesis. Mark 3 is for explaining our control. Leaf 3 has both light and CO2. Now the last 2 marks are really important chain of logic. Because photosynthesis didn't happen in leaves 1 and 2, glucose was not made, and because of that, it couldn't be turned into starch. You've got to hit every single one of those points to get the full 5 marks.
All right, let's keep moving. The next part of the question zooms in on another vital ingredient for photosynthesis, that green stuff, chlorophyll.
So the paper shows you a variegated leaf. You know, one of these cool-looking leaves with green and white patches. They're perfect for experiments because you've got the bits with chlorophyll, the green parts, and bits without it, the white parts, all on the same leaf. It's like having a built-in control group.
So the first question is just to fill in this table. Super simple. It's a real test of the core concept. The mark scheme is dead simple. For the 1 mark, you just need to say the green part will have starch and the white part won't. Easy peasy.
Okay, now for the explanation. How do we get the 2 marks for explaining why? Well, the first mark is for stating the obvious. The green part has chlorophyll, the white part doesn't. The second mark comes from connecting that fact to the process. Because there's chlorophyll, the green part absorbs light, which means photosynthesis can happen and starch can be made. It's that logical link they're looking for.
Next up, a couple of quick-fire 1-mark questions. These are all about knowing your terminology. First, if a plant's leaves are yellow, what ion is it missing? The answer is magnesium. And second, what's the scientific name for that yellow color? That one-word answer is chlorosis. See? Sometimes it just comes down to remembering that one key term.
Okay, let's switch gears. We're moving on from the basic principles now and into data interpretation. This is all about the rate of photosynthesis and the things that can limit it.
So the exam gives you this graph. And whoa, there's a lot going on here. It shows how light intensity affects the rate of photosynthesis under 4 different conditions. The next few questions are all about pulling information from this graph, so you really have to pay attention to what each of those lines represents. First up, 2 more definition questions. Question 1, how do you actually measure the rate? The mark scheme wants a practical answer. You could either measure the volume of oxygen made in a set time, or you could just count the bubbles in a set time. Simple. And question 2, what is a limiting factor? The definition you need is this. It's a factor that, if you increased it, would increase the rate of the reaction. That exact phrasing is what gets you the mark.
Now, for another big one, a 4-mark explanation of the graph. To get all 4 marks, you have to make 4 totally separate points. First, just describe a pattern you see. When you increase the temperature but keep CO2 the same, the rate goes up. Second, explain the science behind it. Higher temperature means more kinetic energy for the molecules, or more enzyme activity. Third, explain the other variable. More CO2 means more substrate for the reaction. And finally, you have to explain why the lines flatten out. It's because all the rates plateau when something else becomes the new limiting factor. Hit those 4 points and you've got it made.
We are on the home stretch of question 7 now. The very last part takes a little detour into physics, actually testing your knowledge on a really important rule about light intensity.
The question is multiple choice. Which of these formulas shows the inverse square law? Basically, this law just says that as you get further away from a light source, the light gets a lot weaker really fast. So take a second. Which of these formulas shows that relationship?
And there it is. Light intensity is proportional to 1 over the distance squared. It's a pretty straightforward mark if you've remembered the formula, and a great little reminder of how all the sciences are connected. So, we have picked apart every single part of this massive question. We've seen exactly what the examiners are looking for. Now let's just pull it all together and think about the main takeaways for doing well in your exams.
And let's not forget this number, 17 marks. That is a huge chunk of a GCSE paper. Getting a question like this right could literally be the difference between 2 grades. So mastering these big, multi-part questions is absolutely key.
So what's our game plan? First, keywords are king. You have to use words like photosynthesis, chlorophyll, limiting factor. Second, when you see a big multi-mark question, think in bullet points. What are the 4 or 5 different things they want me to say? Third, always link your answer back to the data they give you. And finally, know your command words. Explain means why. Describe means what. Make every single word you write fight for a mark.
And we'll leave you with this to think about. Now that you've had a peek behind the curtain, now that you've seen how an examiner actually thinks, how is that going to change your approach? The next time you sit down with a practice question, what's the first thing you're going to look for?